Welcome to the Deep Dive. We're here to bring you the critical insights you need, directly and efficiently.
Hello.
Today we're doing a translational deep dive into a really fascinating area, antisense oligonucleotides. You'll often hear them called ASOs.
Yes, and they represent a pretty fundamental shift, I think, from how we've traditionally thought about drug development.
How so?
Well, for years, maybe decades, the focus was mainly on proteins, right? Finding small molecules or antibodies to interact with a protein, maybe inhibit it, maybe activate it.
Standard pharmacology, yeah.
Exactly. But ASOs, they operate upstream. We're talking about directly targeting the RNA molecules transcribed from genes.
So you're controlling the message itself, not just the final protein product.
Precisely. Directly regulating disease-causing genes or specific variants of those genes right at the RNA level.
Yeah.
And while the idea isn't brand new, the basic science traces back to, believe it or not, 1978.
Oh, that far back.
It is. But the clinical impact, the actual approved drugs changing lives, that's largely been a story of the last, say, 10 to 15 years. The acceleration has been remarkable recently.
Okay, so our mission today is to really unpack that for you, our audience of researchers and clinicians. We want to provide a science-first look.
Right. No hype, just the data and the mechanisms.
We'll cover the core science, how these ASOs actually work inside the cell. We'll look at the chemical engineering, the modifications that were absolutely essential to make them viable drugs.
And we can't talk about ASOs without discussing the approved therapies. We'll go through key FDA approvals, mentioning the dates, the specific diseases they treat.
Definitely. And we also need to tackle the challenges. Delivery is a big one, right? Getting these molecules to the right tissues, and also safety considerations.
Absolutely. Pharmacokinetics and tissue targeting are crucial parts of the story.
So let's start with the basics. What exactly is an ASO?
Okay. Fundamentally, an ASO is a short string of synthetic nucleic acid bases. Think of it as a small piece of DNA or RNA, usually single-stranded.
How short are we talking?
Typically in the range of 15 to 25 nucleotides. And it's important to distinguish them from, say, siRNAs, small interfering RNAs.
Right. siRNAs are double-stranded, work through a different pathway, the RASC complex, mostly in the cytoplasm.
Exactly. ASOs are single-stranded, and their mechanisms can play out in the nucleus or the cytoplasm, depending on the specific ASO design and target.
So the ASO gets into the cell, it finds its target RNA sequence through Watson-Crick base pairing, and then what happens? That's where the different mechanisms come in, based on the ASO's chemistry.
Correct. The sequence dictates where it binds, but the chemical makeup dictates what happens next. There are three main ways they work after they hybridize to the RNA target.
Okay, mechanism one.
The first, and maybe the most classic, is triggering RNA degradation via an enzyme called RNase H1.
RNase H1. That's an endogenous enzyme, right? Already present in our cells.
Yes, exactly. It specifically recognizes and cleaves the RNA strand of a DNA-RNA hybrid duplex.
So for this mechanism to work, the ASO itself needs to have a DNA component.
Precisely. If the ASO is, or contains, a stretch of DNA bases, when it binds to the target messenger RNA, it forms that DNA-RNA hybrid. RNase H1 gets recruited, cuts the mRNA, and boom, the mRNA is degraded before it can be translated into protein.
So it's a direct knockdown of gene expression. You lower the amount of the target RNA.
That's the goal. And we see this mechanism in several approved drugs. Fomiversin, the very first one, used this. Mipomersin, inodersen too.
Okay, that's clear. Knockdown via RNase H. What's the second major mechanism?
The second approach is quite different. It doesn't aim to destroy the RNA. Instead, it modulates RNA splicing.
Splicing. So this happens in the nucleus on the pre-mRNA before the introns are removed.
Correct. Here, the ASO acts via a steric hindrance. It physically gets in the way. It binds to specific sites on the pre-mRNA that are important for regulating splicing. Could be splice sites themselves or enhancer or silencer sequences.
And by blocking those sites.
It changes how the splicing machinery processes that pre-mRNA. And critically, for this to work, you don't want RNase H activation.
Right, because you want the RNA to survive, just spliced differently.
Exactly. So these ASOs are chemically modified, often throughout their entire structure, specifically to prevent RNase H1 from recognizing or cleaving the RNA strand.
And this splicing modulation can lead to different outcomes, can it, depending on what you block?
Yes, two main outcomes we see in therapies. One is exon inclusion. The famous example here is nusinersen, or Spinraza, for spinal muscular atrophy.
SMA, right. Caused by deficiency in the SMN protein.
Correct. Most patients have a backup gene, SMN2, but it normally produces very little functional protein because exon 7 gets skipped during splicing. Nusinersen binds to the SMN2 pre-mRNA in a way that forces the splicing machinery to include exon 7.
Leading to more full-length functional SMN protein, a really elegant solution.
It really is. The other outcome is the opposite, exon skipping.
This is what's used for Duchenne muscular dystrophy, or DMD.
Yes, for certain mutations in DMD. The goal there is to skip over a specific exon containing a disruptive mutation. By forcing the machinery to skip that exon, you can sometimes restore the reading frame of the dystrophin gene.
So you end up with a shorter dystrophin protein, but one that's hopefully partially functional, rather than none at all.
Exactly. And we have several approved ASOs for DMD that do this, like eteplirsen, golodirsen, viltolarsen, casimersen, each targeting a different exon to skip, depending on the patient's mutation.
Okay, so RNase H knockdown, splicing modulation. You mentioned a third mechanism.
The third is also based on steric hindrance, but it targets translation directly.
So acting on the mature mRNA in the cytoplasm.
Yes. The ASO binds to the mRNA, typically near the start codon or in the 5' untranslated region, and physically blocks the ribosome from initiating or progressing along the mRNA. It just stops translation in its tracks.
Simple physical blockage.
Conceptually simple, yes. But it's interesting, as you pointed out in the outline, we don't currently have any approved ASO drugs that rely solely on this mechanism.
Which suggests maybe it's harder to achieve pharmacologically, or perhaps the other mechanisms offer better therapeutic windows.
That seems likely. Perhaps achieving sufficient intracellular concentrations for sustained translation block is tougher, or maybe the RNase H or splicing routes are just more efficient or potent in practice.
Right. Now, none of these mechanisms would matter clinically if the ASOs themselves weren't stable or couldn't be delivered. You mentioned the chemistry being key. Let's talk about those breakthroughs.
Absolutely. The raw, unmodified DNA or RNA oligonucleotides are degraded incredibly quickly by nucleases in the body. They just wouldn't last long enough to do anything.
So modification was essential from the start. What was the first big step?
The first major breakthrough, really enabling the field in the late 80s and early 90s, was the phosphorothioate, or PS, backbone modification.
PS backbone, what does that involve?
It's a simple but profound change. You replace one of the non-bridging oxygen atoms in the phosphate backbone of the nucleic acid with a sulfur atom.
Okay, oxygen out, sulfur in, what does that achieve?
Two critical things. First, it dramatically increases the molecule's resistance to degradation by nucleases, gives it a much longer half-life in biological fluids. Second, it enhances binding to plasma proteins like albumin.
Which affects its distribution and pharmacokinetics.
It does. And crucially, the PS modification still allows the DNA-containing regions of an ASO to support RNase H1 activity, so it provides stability and retains that key degradation mechanism. It was foundational.
That's the first generation. Then came the second generation.
Yes, second gen focused mainly on modifying the sugar part of the nucleotide, specifically the 2' position of the ribose ring.
Like 2-methoxyethyl, the MOE modification.
Yeah.
Or 2-methyl.
Exactly. Those 2' modifications do a couple of things. They further increase nuclease resistance, but maybe more importantly, they significantly increase the binding affinity of the ASO to its target RNA.
Tighter binding.
Higher affinity means potentially more potent effect, or maybe you can use lower doses.
Potentially, yes. And these second-gen modifications led to a very clever design strategy called the gapmer.
Gapmer, explain that. Okay, imagine the ASO strand. A gapmer has a central segment, the gap, made of DNA bases, often with the PS backbone modification. This central gap is what's recognized by RNase H1.
So that's the active region for triggering RNA degradation.
Right. But flanking the central DNA gap on both the 5' and 3' ends are short stretches of nucleotides that have those second-generation sugar modifications, like 2-MOE.
Ah, so the ends, or wings, are highly modified for stability and binding affinity.
Correct. These modified wings protect the internal DNA gap from exonucleases, and their high affinity helps the ASO bind tightly to the target RNA. But because the central gap is DNA, it can still recruit RNase H1 once bound.
That's elegant. You get the best of both worlds. The stability and affinity from the second-gen wings and the RNase H activity from the first-gen psDNA gap.
Precisely. It's a very effective design for ASOs intended to work via RNA degradation. Many successful ASOs use this gapmer structure.
Okay, so the chemistry solves stability and enables the mechanism, but you still have to get the drug to the target cells. That's the PK challenge, right?
Huge challenge. These are relatively large molecules, and they carry a strong negative charge due to the phosphate backbone. They don't just slip across cell membranes easily like small molecules.
So passive diffusion is limited. You need active uptake mechanisms.
Largely, yes. And you generally need to administer them parenterally, intravenous, or subcutaneous injection. Oral delivery just doesn't work well because of degradation in the gut and poor absorption.
Once they're in the bloodstream, where do they tend to go?
For the PS-modified ASOs, which bind quite a bit to plasma proteins, they distribute fairly broadly but tend to accumulate most significantly in the liver and the kidney.
Liver and kidney. Why there?
High blood flow, fenestrated endothelium in the liver cytosolids allows easier access, and filtration processes in the kidney lead to accumulation in proximal tubule cells.
Okay.
Those are the primary organs for uptake and also potential toxicity, which we'll get to.
What about half-life in tissues? You mentioned stability improvements.
Yes, the half-life can be quite long, especially with the second-gen modifications. While plasma half-life might be hours to a couple of days, the tissue half-life, particularly in liver and kidney, can range from maybe 2-3 days for basic PS ASOs up to several weeks, even months, for heavily modified ones like the 2-MOE gapmers.
Weeks to months. That allows for less frequent dosing.
It does. Weekly, monthly, or even quarterly injections are possible for some ASOs, which is a big advantage for chronic diseases.
Okay, so systemic delivery gets you to the liver and kidney well. But what about other tissues? Or avoiding systemic exposure if you don't need it?
Right. That leads to specific delivery routes designed to bypass barriers or target specific compartments. We basically see three main strategies in approved drugs.
What's the first?
Local ocular delivery, right into the eye. The very first approved ASO, fomaversin, used this.
Fomaversin, approved August 26, 1998. That's a key date. For CMV retinitis in AIDS patients.
Exactly. An intravitreal injection puts the drug directly into the vitreous humor, bypassing systemic circulation and getting high concentrations in the target tissue, the retina. And interestingly, that was achieved with just the first-gen PS chemistry.
Shows the power of local delivery when the target is accessible. What's the second route?
Local delivery to the central nervous system, CNS. For diseases affecting the brain or spinal cord, systemic ASOs don't cross the blood-brain barrier effectively.
So you have to go directly into the cerebrospinal fluid, CSF.
Correct. Intrathecal injection, usually via a lumbar puncture. Nusinersen for SMA is the prime example here, delivered right into the CSF to reach the motor neurons.
That requires repeated lumbar punctures, which isn't ideal for patients, especially kids.
It's definitely a burden, and that's driving innovation. You mentioned Biogen's acquisition of Alcyone Therapeutics, announced September 18th, 2025. That's for the Thecaflex DRX system.
What is that?
It's basically an implantable device, a subcutaneous port connected to a catheter that goes into the intrathecal space. The idea is to allow repeated CSF drug delivery without needing repeated spinal taps. A big potential improvement for patient experience with drugs like Spinraza.
Okay, makes sense. Ocular, CNS. What was the third major route or strategy?
It's enhancing systemic delivery, but specifically targeting the liver. Since the liver naturally takes up a lot of ASO anyway, researchers developed ways to make that uptake even more efficient and specific. The breakthrough here was GalNAc conjugation. N-acetylgalactosamine, GalNAc, is a sugar molecule that binds very specifically to the acetylglycoprotein receptor, or ASGPR.
And that receptor is primarily found where?
On the surface of hepatocytes, the main liver cells. Massively abundant there. So if you attach GalNAc ligands to your ASO...
The ASO essentially gets actively pulled out of the bloodstream and into liver cells via this receptor.
Exactly. It dramatically increases the potency of the ASO in the liver, sometimes by tenfold or more, allowing for lower doses and potentially reducing exposure and toxicity in other tissues like the kidney. It's been a huge enabler for liver-targeted ASO therapies.
Okay, these chemical and delivery innovations seem to have really unlocked the clinical potential. Let's look at that timeline you mentioned. A slow start, then an acceleration.
Very much so. Fomaversin in 1998, then a long gap. Only 2 more ASO approvals over the next 18 years. Mepomersin was one in 2013.
But then things picked up.
Markedly quickened, yes. From 2016 through 2021, we saw 7 new ASO drugs approved by the FDA, a real validation of the platform.
Which ones were the real game changers in that period?
Well, nusinersen Spinraza has to be high on the list. Approved December 23, 2016 for spinal muscular atrophy.
First drug ever for SMA, right? A devastating disease.
Absolutely. And as we discussed, it works via exon inclusion of SMN2 delivered intrathecally. The clinical data was compelling. The pivotal ENDEAR trial showed a 47% reduction in the risk of death or needing permanent ventilation compared to the sham control. Really changed the outlook for patients and families.
A landmark approval. What else stands out?
The portfolio for Duchenne muscular dystrophy, DMD. This uses that exon skipping mechanism we talked about.
Right, but these use a different chemistry, don't they? Not the gapmers.
Correct. The DMD exon skipping drugs, eteplirsen, golodirsen, viltolarsen, casimersen, use a chemically distinct backbone called PMO, which stands for phosphorodiamidate morpholino oligomer.
PMO chemistry. How is that different?
It's quite different from the PS backbone. PMOs are uncharged molecules, unlike the negatively charged PS ASOs. And critically, their backbone structure is not recognized by RNase H1.
Ah, so they are inherently incapable of causing RNA degradation via RNase H.
Exactly, which makes them ideally suited for mechanisms like splicing modulation or steric block, where you don't want the RNA target destroyed. They primarily work in the nucleus on splicing.
Okay, so the PMO chemistry is tailored for the exon skipping job in DMD. Let's list those approvals again.
Sure, eteplirsen came first in 2016, targeting exon 51, then golodirsen in 2019 and viltolarsen in 2020, both for exon 53 skipping, and casimersen in 2021 for exon 45.
It's quite amazing. 4 different drugs, each tailored to skip a specific exon, matching different patient mutations, shows the precision possible.
It does highlight the modularity, but it's important for clinicians and researchers listening to remember that these DMD drugs received accelerated approval.
Based on a surrogate endpoint, right?
Yeah.
Increased dystrophin protein levels in muscle biopsies.
Correct. Not initially based on definitive proof of improved clinical function like walking ability. Demonstrating clear clinical benefit has been more challenging, and confirmation is still pending or ongoing for some. It's a point of discussion in the field.
Understood. Any other major milestones from that 2016-2021 period?
Yes, anodersen, Tixedi, approved October 5, 2018. This one is for hereditary transthyretin amyloidosis, or HATTR.
HATTR amyloidosis, a progressive debilitating disease. How does anodersen work?
This one goes back to the first mechanism we discussed. It's an RNase H-dependent ASO, a gapmer actually, that targets the mRNA for the TTR protein transthyretin.
So it knocks down the production of the TTR protein, which is what misfolds and forms amyloid deposits in this disease.
Exactly. Reduce the protein production, reduce the amyloid buildup.
Okay, so a growing list of approvals tackling serious genetic diseases. But ASOs aren't without risks. We touched on liver and kidney accumulation. Let's talk about the common safety concerns, the class effects.
Yes, this is critical for anyone working with or prescribing these drugs because many ASOs share similar chemistries, particularly the PS backbone, and similar distribution patterns. They can share certain toxicity profiles.
What's the first major one?
Hepatotoxicity or liver injury. This is a known risk. We often see elevations in liver enzymes, ALT and AST, in patients treated with some ASOs. Sometimes hepatic steatosis or fatty liver can also occur.
How serious can this get?
It can be very serious. Mipalmersen, which was approved for homozygous familial hypercholesterolemia, HoFH, back in January 2013.
I remember that one. Targeting ApoB mRNA.
Right. It was actually withdrawn from the market in Europe and later the US around 2019, primarily due to the risk of severe hepatotoxicity, including steatosis. That really highlights the potential severity.
So liver function monitoring is essential.
Absolutely. And Oderson, for example, also carries warnings about this and requires regular liver function tests.
Okay, liver toxicity. What's the second common concern?
Renal toxicity or kidney injury. This has been observed with several ASOs, particularly some of those used for neuromuscular diseases. Nusinersen, inodersen, and the DMD drugs, golodirsen, viltolarsen, casimersen have all had reports.
What's the thinking on the mechanism there?
It's likely related to the drug accumulating in the proximal tubule cells of the kidney during filtration and reabsorption processes. High local concentrations might damage those cells.
So kidney function monitoring is also needed.
Yes, monitoring urine protein levels, often as a urine protein to creatinine ratio, is generally recommended to catch early signs of kidney effects.
Liver, kidney, any other major class warnings?
The third significant one is coagulation abnormalities, specifically thrombocytopenia, which is a low platelet count.
Low platelets increase bleeding risk.
Correct. This has been a particular concern for nusinersen and inodersen. Both carry warnings about this risk.
meaning you need to check platelet counts before starting and during treatment.
Yes, baseline platelet counts and coagulation tests like PT and APTT are needed, along with regular monitoring before each dose for drugs like Inodersen. It's a manageable risk with monitoring, but a serious one.
Okay, so significant efficacy in tough diseases, but balanced against real risks needing careful management. Where is the field heading now? Are ASOs moving beyond just knockdown and splicing?
Absolutely. The platform is proving quite versatile. We're seeing expansion into new types of mechanisms and disease areas, especially in preclinical research.
Like what?
Well, there's work using ASOs to target pathways involved in things like oxidative stress or metabolic diseases. One example mentioned in the sources was using ASOs against a target called MST3.
MST3, what did that do?
In mouse models of non-alcoholic fatty liver disease, NAFLD, these MST3 ASOs seem to help by reducing liver steatosis and inflammation, so targeting metabolic signaling pathways.
Interesting. Moving beyond rare genetic disorders towards more common metabolic conditions.
Potentially, yes, though that's still early stage. But perhaps even more exciting is the development of entirely new types of ASO mechanisms.
You mean besides knockdown, splicing, and translation block?
Yes. One really innovative approach gaining traction is called TANGO,
Tango, what does that stand for?
targeted augmentation of nuclear gene output.
Augmentation. So increasing gene expression. That's the opposite of knockdown.
Exactly, it flips the script. Instead of silencing a bad gene or fixing a splice defect, Tango uses ASOs to upregulate the expression of a normal wild-type protein.
How on earth does that work?
The strategy often involves targeting specific sequences, sometimes called poison exons or inhibitory regulatory elements within the pre-mRNA, or even non-coding RNAs, that normally act to suppress the expression or stability of the target transcript, by using an ASO to block these negative regulators.
You release the brakes, allowing more functional protein to be produced from the gene.
Precisely. It's a really clever way to boost protein levels.
What kind of diseases would that be useful for?
It's particularly promising for diseases caused by haploinsufficiency. These are conditions where having only one functional copy of a gene instead of the usual two doesn't produce enough protein for normal function.
So you use Tango to boost the output from the one good copy the patient has.
That's the idea. The source material mentioned preclinical work using this approach for Dravet syndrome, a severe form of epilepsy often caused by haploinsufficiency of a sodium channel gene. Using ASOs to block a non-productive splicing event to increase the functional channel protein.
That sounds potentially transformative for a whole class of genetic disorders.
It could be. It really broadens the scope of what ASOs might be able to treat.
But all these exciting new applications, whether it's metabolic disease or Tango, still run into that fundamental challenge we discussed earlier, right?
The delivery challenge, yes.
Getting the ASOs efficiently and safely to tissues beyond the liver, kidney, eye, and CNS.
That remains the major hurdle, the key bottleneck for really expanding the therapeutic reach of ASOs. Developing technologies for effective extrahepatic delivery, getting functional amounts into muscle, bone, the heart, maybe specific immune cells, that's where a huge amount of research effort is focused now.
Because if you can solve that, the number of potential disease targets just explodes.
Absolutely. Cracking extrahepatic delivery is probably the single biggest goal for the next phase of ASO development.
Okay, let's try to wrap this up. It's been a fascinating journey.
It really has. ASOs have come such a long way. From that initial concept back in '78 through decades of painstaking chemical refinement, the PS backbone, the 2 mods, the GAPMers, the PMOs.
Leading to sophisticated molecules that can precisely target RNA to either knock it down via RNase H or modulate its splicing, or potentially even augment its output.
And resulting in genuinely life-altering drugs for previously untreatable conditions, like nusinersen for SMA, real clinical breakthroughs.
But always shadowed by the constraints of pharmacology, the delivery challenges dictating administration routes, and the accumulation in liver and kidney driving key toxicities that need careful management.
That balance between efficacy and safety, mediated by chemistry and delivery, is the core story.
Which brings us to a final thought, something for our listeners to consider. It relates to how we measure success, especially with these powerful targeted therapies.
You're thinking about the tofersen approval.
Exactly. Tofersen, an ASO for a specific genetic form of ALS, SOD1 ALS. The FDA granted accelerated approval in April 2023.
But the phase 3 trial hadn't actually met its primary clinical endpoint, right? It didn't significantly slow functional decline compared to placebo over the main study period.
Correct. The approval was largely based on a biomarker. Tofersen significantly reduced levels of neurofilament light chain, NFL, in the CSF, which is thought to be a marker of neuronal damage.
So the drug clearly hit its molecular target and changed a relevant biomarker, even if the clinical benefit wasn't definitively proven in that time frame.
Right. And it raises a huge question. As ASOs and other genetic medicines get better and better at precisely modifying specific RNA or gene targets, and we can measure those changes with biomarkers, how do we, as a clinical and regulatory community, weigh that molecular evidence against traditional clinical endpoints?
Especially in devastating diseases like ALS, where clinical trials are difficult and time is critical.
How much molecular correction is enough to warrant approval, even if the downstream clinical effects take longer to manifest or are harder to measure conclusively? Where do we set the bar for defining therapeutic success versus acceptable risk?
It's a profound question. Does hitting the biological target precisely, as measured by a biomarker, become a new standard of evidence, at least for accelerated approval in severe conditions?
It forces a reevaluation, doesn't it? The increasing power of these ASO technologies is pushing the boundaries not just of therapy, but also of how we define and regulate therapeutic value itself.
A challenging but crucial conversation for the field moving forward.
Indeed. Something important for all of us in this space to keep thinking about. Thank you for that insightful deep dive.
My pleasure. It's an exciting field to watch.
And thank you all for joining us on the deep dive.